Okay, so hello everyone. Welcome to the WinS Inspiring Scientist session with Dr. Chelsea Reichert Plaska. She's a former fellow grad from CUNY. She was a student in my program at neuroscience at the Grad Center, and her lab was located on CCNY's campus. She just defended her thesis in April 2021. So in part, her grad career was and dissertation was during the pandemic. In her talk, she'll discuss not only what it's like to be a recent graduate from CUNY, but also what her research journey was like before and during the pandemic. Her dissertation examined the neural correlates of working memory maintenance of complex visual stimuli using high-density EEG. During her time at CUNY, she focused on learning neuroscience techniques, including EEG, simultaneous EEG and fMRI, and eye tracking with the goal of applying these techniques in a clinical setting. She took a postdoctoral research position at the geriatric psychiatry division at the Nation S. Klein Institute, which is a clinical research laboratory that studies aging and cognition. Her current research seeks to elucidate the factors that impact cognitive change to better understand and predict individuals that are likely to transition to Alzheimer's and related dementias. So she has prepared a really interesting talk about research and our journey at CUNY and beyond. And on that note, Chelsea, the floor is yours.
Thanks, Naomi, and thanks everyone for having me. Let's see if I can share my screen. You have to give me the option to share my screen.
Okay, I just changed it.
Okay, cool. Awesome. So yeah, so as Naomi said, can everyone see this? I'm assuming, yeah. Okay, minimize this. So as Naomi said, I actually just finished my PhD in April. And so I'm currently doing my postdoc at the Nathan Klein Institute. And I figured I'll give you a little taste of the research that I did during graduate school and spend some time talking about what I'm doing now. Naomi says that some of you guys are from the med school, some of you are grad students. So I have a little bit of both, since a lot of what I do now is very clinically focused. But first, I wanted to give you a little insight into my journey because it was kind of twisted in my opinion. And I think maybe some of you might understand what I've gone through and are going through the same. So I completed college a long time ago. I was a biology major, like many biology majors. I was pre-med. And during my pre-med journey, I was trying to diversify myself in application terms. And so I joined a research lab, my first research experience actually, and it was called the Alcohol Lab at College of New Jersey. And it was a behavioral research lab that looked at animal models of alcohol abuse. And it was a great introduction into research. I really liked the topic. I like studying mental health. I like studying alcohol behavior, but I really hated working with animals. It's not for everybody. And so I knew immediately that that was not something for me. But I did enjoy reading articles. I did enjoy lab meetings. And so I stuck with it for about 3 years. And after TCNJ, as I was furthering my education and trying to make myself a better med school candidate, I continued on to Seton Hall University to do a master's in experimental psychology. As I mentioned, it was to make myself a better med school candidate. And that's because at the time of graduation, I was not ready to apply for med school. My MCATs were not high enough. So I thought that this, again, would help me to become a better candidate. While at Seton Hall, I actually started out in a general psych program, which was way too general and a little bit too clinical focused. And so by the time I switched into an experimental psychology program, I didn't really have a choice of lab. And so I ended up in a social psychology lab, which is interesting. We studied mental health issues and specifically stigma related to mental health issues. So I was able to keep studying alcohol dependence in that framework. And I also looked at depression. Very interesting. Again, I enjoyed the lab meeting part. I enjoyed learning about how to build experiments and how to analyze data and write papers, but I knew that that topic was just, I don't know, it almost seemed a little too logical. And so I wanted to study something a bit more science focused, a bit more tied to that biology degree I had. but I didn't quite know what I wanted to study yet. So I actually started working after my master's, and I ended up at the Nathan Klein Institute, where I am today, in the exact same lab that I am in today, which is the Geriatric Psychiatry Division. So at this lab, we study aging and cognition in a population that's about 50 and older. We do study Alzheimer's disease and related dementias, but a lot of the people we work with are normal, and we're trying to early diagnose any sort of impairment that could lead them to Alzheimer's disease. And of course, I promise to get into that, but that's just a little taste of the overall focus of the lab. And when I was hired way back in 2012, I was hired as a neuropsychological rater. I was not at all interested in memory or cognition at the time. I was just there to do a job and get some experience. But after about a year of working in the lab and working with our patients, testing thousands of people actually, and doing clinical trials, I really fell in love with the work. And in particular, I was interested in the heterogeneity of the cognitive profile. So what I mean by that is on paper, you can have somebody who is, testing impaired, having problems with memory, having problems with attention, something that might be considered necessary for medication. But when you meet the person and you look at their psychiatric profile, you look at their mental health profile, you'll actually see that they're normal. They're just testing poorly. Or on the flip side, you'll have all these patients with the same diagnosis and then you test them and they all seem very different. So to me, that was fascinating. And that inspired me to go back to school and help me to decide that I definitely wanted to study memory, definitely wanted to study cognition, but I wanted to understand what was going on in the brain. And so that brought me to a PhD in neuroscience. And as Naomi mentioned, I went to the Graduate Center. I was in the Behavioral and Cognitive Neuroscience program. And like I said, I wanted to study memory, but at CUNY, there was only two, maybe three labs that were really studying memory and also doing some sort of neuroscience technique that would allow me to study the brain. So EEG, functional MRI. One of those labs was not taking students. The other one was potentially taking students, which brought me to the Memory and Cognition Lab at City College. So that is really where I got a lot of my experience. Here's just a picture of all my awesome lab mates that I spent a few years with. Let's see. So during that time, I actually went in the lab to learn MRI. And unfortunately, due to CUNY and all sorts of, stuff that was out of my control, the MRI center was delayed by 4 years for setting up. So even though I was told in 2015 when I started that I was going to be learning MRI, I was not learning MRI. Instead, I learned EEG, which is what these two pictures are here. So here's me training some of our undergraduate students, to put on an EEG cap, gel the cap, properly place the electrodes, so on and so forth. So that is how I spent my time from 2015 for about 3 years into the program. While I did have a lab manager there who was in the lab before I started, I was the most senior person as a graduate student. So he did show me how to start collecting EEG, but much of what I learned or much of what I did in graduate school was self-taught. So I spent all of my time figuring this stuff out, how to best collect EEG data, how to process it, how to analyze it, how to use the programs that we did. And of course, this is all while teaching due to, funding issues, whatnot. I did spend some time in another lab at Baruch, which was an attention-based lab that also did EEG and MRI. I really love that lab too. And I got along very well with the mentor, but unfortunately, due to funding, she was not able to take me. So I stayed at City College. And so jump right into some of my research. After all of that time spent learning EEG, I actually did secure some funding from a graduate student grant to do 2 studies. And this is actually the pilot study for those studies. And so, and that actually led to my first publication. But before I talk about that, let me just tell you a little bit about the type of research that we did in our lab at City College. So the focus was understanding the neural correlates, of working memory. And specifically, we were interested in maintenance. So I'm going to direct your attention to the right side of the screen. So this is an example of a task that we do when we did EEG. And this is a working memory task in particular. So first you would show images or words or sounds and have people try to learn the information or encode it, followed by a period of time where they're not looking at anything, not seeing anything, not hearing anything, called the delay period. And this is when maintenance occurs. And so this is the period of time that we were most interested during a lot of our studies, followed by a period of time where they recall the information and you measure how much they did retain or not. And so for the sake of time, what we did is we looked at a 6-second period during which information was maintained. And we looked at the EEG signals during that time period to better understand how people are maintaining information and what areas of the brain are involved. And so down here on the right are some plots of that 6-second period of time. Time is on the x-axis. On the y-axis is frequency. So we don't just look at the signal across time, we look at the signal in terms of frequency. And so what we found was, and this is going to seem kind of like a duh type of finding, is that the pattern of activity is transient. So this red pattern here represent increases in activity early in the delay period when people are maintaining information, followed by a pattern of deactivation later in time. And the second plot here actually has an overlay of correlations with performance. So the more intense or the greater increase in activity, the better people perform. So there is a positive correlation. On the flip side, in the second half of the delay period, when the activity is becoming more deactivated, people are actually doing worse in performance. So it's a negative correlation. So this pattern, this transient pattern, was kind of perplexing to us. It didn't really sit with the existing literature. And so the projects that I worked on and the grant that I got was to better understand, why are we seeing this transient pattern? And so in my mind, I thought, well, when somebody's maintaining information, what are they doing? So to me, they were rehearsing. Now, not everybody is actually rehearsing, I've come to learn. So what we did is actually manipulate this. So this is an experiment from my dissertation, and this is actually from my first publication, which, side note, took 2.5 years to publish. It was a very, very trying process. But so this is a very similar working memory task. Some differences to note is that here, instead of seeing complex scenes that are very easy to label or to identify what's inside them, we actually showed phase scrambled scenes, which are a little more difficult to label. They actually lack semantic content. So what we had people do was be creative, come up with a label for it. And then during the delay period, when you're trying to maintain that information while you're thinking about it, we actually have you rehearse or repeat that label over and over again. So we're trying to help you remember that information. In the other condition, we had not a control, but we actually had people not rehearse. So we had them suppress. So when they looked at the images, they didn't do anything except look at them. And then during the delay period, we had them repeat the word the over and again, which in the behavioral literature shows that people won't rehearse if they're doing that. And perhaps not surprisingly, people who rehearsed did better than people who didn't rehearse. And oddly enough, there were no EEG experiments that had looked at that. And so one of the findings was that, yes, we did find that transient pattern of activity that we saw in our earlier experiment. So that could explain that when people were maintaining those normal complex scenes, they were rehearsing, and that's the pattern we saw. I'm not showing that here for the sake of time. Instead, I'm just showing you a brief slide here that shows you not just the delay activity, but what is the source of the delay activity. So a problem with EEG is that there's volume conduction issues, and so the actual source is hard to find. So a lot of what we look at is just surface activity, but we can use models of the brain to try to figure out what is the source. And so here, what we did is we applied a source montage of 15 discrete brain regions, and we compared the activity during the rehearsal condition to the activity during the suppression condition. And the reason for that is because participants were doing better when they were rehearsing. So we wanted to see what was the source of that activity, what could possibly be supporting its performance. And so what we identified in this analysis is that the left anterior temporal region, which is shown here on this little brain schematic, was the source of the rehearsal activity. And it was specifically found in 2 of the lower frequency ranges, which is the theta and alpha band. So greater increases in theta and greater increases in alpha from the suppression to the rehearsal condition was associated with better performance. Again, maybe not all that surprising, because that's, language related areas, could also be related to memory or some sort of association between the semantic label and the visual information, so on and so forth. Okay, I know I'm flying through all this, so if there's any questions, I am happy to answer them at any point. Um, but so I completed those 2 experiments, collected the data, wrote up the paper, went through 5 or 6 journal submissions. 2.5 years later, finally got the paper published the month before my dissertation, which was a miracle. And as I mentioned a few minutes ago, my original goal was really to do MRI. So in 2018, I believe the scanner at CCNY finally arrived. And so it was kind of a unique experience because myself and some of the other graduate students were able to be there as the center was being set up. And of course, finally getting experience collecting MRI data. So I'm not showing anyone here. This participant is actually my husband, so we have permission to show him. And here is a sagittal slice of uh, a T1 scan of his brain. So yes, I started MRI, but we also got experience doing simultaneous EEG fMRI, which is really complicated, but very cool to do. And the unique thing about my experience was that since no one had done it before, I was kind of involved in the setting up of not only this technique, but understanding how to best collect the data, how to analyze the data. I created a pipeline for processing and even was teaching or working with the MRI department to figure out how to best do this. So I was really excited. And of course, both my experiments and then some of the simultaneous EEG fMRI experiments that we've been collecting I had been analyzing, I was able to present at SFN, which was my first huge neuroscience conference. That was in October 2020. Getting some feedback during that was crucial because then I was able to start designing my fMRI experiment. So we had done these EEG experiments. We had some findings about rehearsal, but we weren't exactly sure where in the brain this was going. And even though we were able to do these source analyses, they were not necessarily as accurate as an fMRI experiment would be. And so that was the plan. That was part of my dissertation. That's what my committee approved. And in fact, where I met Naomi was at the U Michigan fMRI training, which was supposed to help me prepare for this experience. But then COVID happened. So that was March 2020. My MRI experiment was supposed to start in April 2020. So I think it was like March 12th or March 16th, we had an MRI experiment lined up. We did it, and then we found out CUNY was closing and CCNY legitimately closed, as you all know. So this was really nerve-wracking, problematic, all the scary things going on in the world aside, because all of our data was in our lab. We took copies of raw data, but the program that I'd been using for the past 4.5 years was in the lab and couldn't leave the lab. And the process data that I'd been working on was also in the lab. When we shut the lab down, we only thought we'd be gone for a week, a month at the most. And at this point, I've been 4.5 years in and I really wanted to graduate, and it wasn't looking good. So it was tough, but one good thing is that since we were home for so long and we didn't get back into the lab until July 2020, I had the time to learn coding or get better at coding. And so with that, I actually learned how to process EEG data using MATLAB. This is a random screenshot of EEG lab in the background. I also learned MD Python. And so I really focused my time on that because I ended up for the third, or I want to even say fourth time, reprocessing all of the data. And at this point, since it's July 2020, we're just getting back in the lab and we don't even really know when we're going to be collecting data again. My MRI experiment was officially canceled and my committee decided that I would work with data that I already collected. So here is an example of another experiment that I worked on. This was a simultaneous EEG fMRI experiment. And I focused specifically on the EEG data. So it had nothing to do with rehearsal. We did focus on the delay period. So the overall interest is the same, which is understanding what are the neural correlates while you're maintaining complex visual stimuli. Here, this is more of a classic task. It's a load working memory task. So there's 2 images here, a low working memory load versus 5 images, which would be a high working memory load. And then the same 6-second delay period and understanding what goes on during this period of time. The difference between this experiment and the ones that I showed earlier, aside from the fact that there's no rehearsal going on and no suppression, there are 6 phase scrambled images here. And the idea was that these would serve as a phase scrambled baseline period. So instead of looking at a blank screen, you're actually looking at some visual input, but it has no meaning. And so the thought was that people would just do really well on the task. They would ignore these images, and it might not even matter if it's just 2 or 5 images, everyone will do really well. What we found was that people did worse on the high load performance, the load 5 performance, than they did on the load 2 or the 2 image task. That's actually not really surprising. 2 images are easier to remember than 5 images. And so something that we didn't realize at the time is that anytime you put something on the screen, and even if someone's told to ignore it, visual input serves as attentional interference. And so we were trying to understand what the presence of that information during the delay period does to the brain activity. And so to show you just one select result, we use a connectivity analysis of the EEG data, which is a way to understand what brain regions might be communicating during this time period to facilitate performance and block out the interference. And so we compared the low load condition to the high load condition. And what we found is that there was increased connectivity using phase locking between these 3 regions that are highlighted here. So the left frontal to the right posterior temporal, left anterior temporal to the left posterior temporal, right anterior, so on and so forth. And they're also displayed here on these brains. And so what you'll see here is that the activity in each of them continues throughout most of the delay period, and they're isolated in the lower frequency bands of theta, alpha, and beta. And each of these regions we ascribe to the successive performance on the low load was the filtering of the interference, which is likely easier to do when you only have 2 images in mind than when you have 5. Of course, my favorite term, I think rehearsal's going on, which again is easier to do when you have 2 images than not 5. And probably you're better able to control your attention during that time period as well. Okay, so I know that was like flying through some of the results. I had a 300-page dissertation, so happy to answer questions on that. But yes, I did successfully defend in April 2021, 1 year later than planned, but I finished nonetheless. And I'm now at the start of what I hope to be my career. So I did something a little unusual, at least what I've been told, and that is I returned to a lab that I'd already been a part of. And this is something that was strategic on my part, and that is because I really wanted to work with older adults, and I really wanted to study Alzheimer's disease and hopefully use, the neuroscience techniques I learned in graduate school and use them in this lab, which is actually more of a clinically based lab. I work with a lot of MDs and some PhDs, but a little bit different focus. And at the time that I was hired for my postdoc, we were actually just told that we were getting an NIA, an R01, a big grant. And so they absolutely needed me to start ASAP. And so I did start before I even finished my dissertation edits, which was a little challenging, but worth it nonetheless. And so to give you a little insight into what we work on now, so we do cognition and aging, like I said, but we actually also look at late life depression. So depression and Alzheimer's disease are 2 things that are actually related. A lot of people that develop depression later in life, let's say after the age of 60, or even people that develop depression earlier in life, so early onset depression, have a greater risk of developing Alzheimer's disease, than people who don't. So at the very beginning of my time at NKI, we were just finishing up an R01 that was looking at the connection between depression and Alzheimer's. It was an observation study where people were followed longitudinally for 3 years. There was a depressed group, there was a control group. And during the baseline period and the 3rd year follow-up, a lumbar puncture was done to measure cerebrospinal fluid. And lots of biological variables were measured in cerebrospinal fluid to see the changes in these biomarkers and also compare them with controls. And so what I'm showing here on the screen is from an earlier published paper. This is actually not a paper I'm on. So what you see here on the left is HAM-D score. On the right is CSFA beta 42. So CSFA beta 42 is one of these hallmark biological markers that is associated with Alzheimer's disease. It tends to be low in people that have Alzheimer's disease, and the level is related to the amount of beta amyloid plaques that are present in the brain that are ascribed to possibly causing Alzheimer's disease. This graph here on the right, HAM-D, this is the Hamilton Depression Score. Hamilton Depression is an interview-based measure that uses the DSM criteria for depression and measures how severe somebody's depression is. Higher scores mean more depression. So the red line is controls for both graphs. The blue line is depressed for both graphs. And so what you can see at the baseline visit is the depressed group are very depressed. The control group are not depressed. And across the 3-year period, which would be the follow-up, the depressed group actually gets better. Control group stays about the same. And at the same time, you actually see changes in these CSF beta markers. So while the depressed group has lower CSF beta, which is more like people with Alzheimer's disease, as their symptoms resolve, this marker actually starts to normalize. And so this finding is a bit unique, or at least at the time it was unique, and it has served as the foundation for this RO1 that we were just rewarded, that I've actually spent much of my time helping to set up now that I've returned full-time to NKI. And so we just started recruiting, and this is going to be a clinical trial, again, to study the relationship between depression and Alzheimer's disease. It's going to be an 8-week treatment trial in elderly depressed. And what is going to happen during this trial is that currently severely depressed people will be recruited. We're going to wash them off their antidepressants, and then we are going to give them an FDA approved antidepressant for 8 weeks, and we're going to measure the changes in their depression symptoms. And of course, most importantly, we're going to look at differences between their baseline and 8-week CSF biomarkers, CSF beta 42, of course, and a whole litany of other biomarkers that are related to Alzheimer's disease, vascular disease, and depression as well. That. And of course, Naomi, you can stop me at any time you need. This is the other program that I work on, which is called the Mary program. This is a longitudinal project, or program rather, that has been established back in 2003 in partnership with Rockland County. It's this really unique program that has people come in and we collect cognitive data, clinical and psychiatric information. We also collect blood plasma, vitals. We all do all different sorts of surveys on depression, anxiety, lifestyle, state, trait, mood, all sorts of things that you can think about. And we have people come back every single year. So one of the things that I've done, and actually this is one of the things I did during graduate school, is I helped to secure additional funding for this program. This is something that, while it was established in partnership with Rockland County, it lost its funding over the years, perhaps due to lack of interest and not having somebody young around who was really, focused on this program and interested in keeping it alive. And so I helped them to write a grant to tell the county and the world how important I thought this program was, because it is a clinical service that is free to residents. They're volunteers, they do not get paid. The benefit to them is that they get a summary of their performance on the cognitive tests, and we give them recommendations. And that's something that I'm involved with, is determining what is the clinical status of these people. So I look at their cognitive performance, I look at how they perform in comparison to people who are their age and their education level, and I come up with whether or not they're cognitively normal, whether they're prodromal Alzheimer's disease or mild cognitively impaired. So where are they starting to show declines in terms of memory, attention, executive function? Or if they are showing these patterns, is it related to depression? Could it be related to uncontrolled medical conditions? All sorts of things that you can think of. And actually, before I started graduate school, I had the opportunity to write up this program in a book. We wrote it up as a community model that could be applied in any clinical setting. And at the time, I think we had a little over 1,000 people. Here's just a summary of the number of people that have participated and the number of visits. To date, we have over 1,300 people that have come for up to 17 visits, which is really kind of cool. Here is the age range kind of summary. So we see a lot of people in their 60s and 70s and 80s. I tested people as old as 99, which is really cool. And as I mentioned, actually, it was observing the heterogeneity of the cognitive profile of the people in this group. That is the reason I went back to graduate school. And today I'm trying to understand, and I hope to be able to apply machine learning and all sorts of different things to this to better understand how we can predict people transitioning to Alzheimer's disease. What is it about their cognitive profile that is that we can use to better understand this process? Because the classic criteria for diagnosing somebody with mild cognitive impairment is just that they have to be significantly below average on one memory test, maybe two. There's an updated version of this criteria where it's a multi-domain impairment. So you have to be below average on memory and then maybe one other domain like attention or executive function. And you don't necessarily need to think about all the other things that are going on in their world. They should have subjective memory complaints, but they should be functionally normal. And what I've learned in my time here is that there is so much more going on. And there's also so much that we don't capture. And so actually I'm in the process of preparing a grant for the Retirement Research Foundation for this particular program. I actually just wrote a draft of it and I've been sending it to some of my mentors at NKI. And also I have some mentors at NYU Langone that we work with. And so the idea is that we're going to be developing a pilot program to examine social isolation in our population, which has been found to both modulate AD biomarkers, depression biomarkers, as well as cognition. And this is something that has been exacerbated by the pandemic, especially in elderly individuals, and especially those that are less familiar with technology and how to reach out to people during this time when you can't physically see each other. And so I want to capture this. Lots of people have been doing it, but it's quite new. And so we're going to be looking at a 12-week social isolation social engagement program, where we then measure changes in plasma biomarkers. And I hope to then also secure additional funding for MRI, because I think that would be very interesting. And I guess lastly, you might be thinking, well, you went to school for neuroscience, you're now in a clinical world. How do you bridge these two things together? And so, as you can tell, I'm very early in my career, but my plan is to use EEG and MRI, and of course, study this aging population and better understand how we can early diagnose people. And I think some of the things that we're starting to do in the field is to use functional MRI, use functional connectivity, both in EEG and MRI, and apply these techniques to this population, which can be very tricky. There are certainly a lot of people that aren't able to go in MRI because of various medical conditions. Certainly be difficult to bring people with Alzheimer's disease into a scanner, but I think it would be an exciting thing to do, or possibly even work with people who are in the early stages, or their children, or people who are at high risk. There's so many different possibilities. And so that's kind of where I'm at. I haven't even been in my postdoc for a year. So a lot of what I do is really just focused on working on a grant, working on a program that I help to get funding for. And when I have some time, I read articles. And when I have some time, I write a little bit of a grant, so on and so forth. So I hope that wasn't too much rambling. And I, of course, welcome you to email me. I'm happy to share any articles, my publications, some of the work we do in our lab. I, of course, want to thank everybody for my current lab. Dr. Mara is my boss, his wife. She runs our office. She's amazing. My former lab at CCNY, Dr. Elmore is my advisor. Of course, these are all my wonderful lab members, and Rockland County Mental Health and NIH and NAA for funding the various experiments. So I am happy to take questions. Yeah.
Thank you so much for all that beautiful information. I didn't get to introduce myself at the beginning, but I'm also part of CCNY WinS, and I'm so glad I joined this talk today. Honestly speaking, I don't think I've ever heard like someone else explain how they started off as pre-med and then transitioned into research. That's exactly how I started off. I was like pre-med, and then I also wanted to diversify my resume and my experiences. So I got into the Emerson lab at City College, and I fell in love with like the research aspect of science and really like the nitty-gritty science stuff. So that's why I ended up transitioning. But if it's okay with you, I just had a few questions that I wanted to get right into. So yeah,
Sure.
Okay, so I guess my first question would be for me, and I guess hopefully the other ladies can benefit from this. What would be your advice to young women who are moving up the scientific academic ladder in tackling like gender bias and inequality? Because I know like the number of women in science is far less than the number of men in science. So what's your advice for tackling that gender bias that exists in our field?
I mean, it's a great question, and I'm certainly open to anyone's insight into this. Um, I think it's something that we're still working on. Um, I think the best thing that you can do, and I know people have told me this, is to have a female mentor. And it's something that I didn't have in graduate school, I would say, which was really tough. Um, but so that's why it was so important for me now to choose the lab that I did. My boss is a male and his wife runs our office, but they're just very unique individuals. They have daughters. And so I feel like for them, females and males are completely equal. And so it's conversations that we have openly together all the time about my experience in graduate school, other people's experience. They have a daughter that also has had similar experience, had a male mentor. And um, so female mentor, open conversation. And, so I'm at the point now where I'm finding mentors, and I've actually just met with the head of where I work at NKI, and also basically my boss's boss at NYU at Langone Health, and kind of talked about my career trajectory and who my mentors would be. And it's funny that there are no females that they suggested for me. They gave me an imaging mentor and a biological mentor and a computation mentor, and they're all these like established males. But my boss has suggested female mentors. So there is going to be at least one female who will be down the hall from me, who's kind of like in her late 30s, early 40s, who actually just got an R01. And so he personally has to connect me with females who I know have had similar experiences, who I can talk to. And I think that's the best advice I have. I haven't followed it yet because it's also tough. You want to be able to open up to them, and so it's, if you can find someone that you think has had a shared experience that you feel comfortable with, that's I'm sure the best thing that you can do.
Thank you. Yeah, I think that like having a female mentor would definitely, like, I know in my case, I don't have any female mentors either. But luckily the first lab that I was in, the Emerson lab, I know for sure Dr. Emerson is like a feminist. Like he would go to bat for all of us. I think his lab was a majority of women. And so I like had like one experience where there was like a microaggression by like a lab next door by a male, and I brought it up to his attention and he immediately addressed it. Like he didn't let a minute go by without addressing it. And so it made me feel really seen, and I didn't feel brushed away. So, but definitely I think a female mentor will understand like at a different level. But thank you so much for answering that question. And then
I'm going to pipe in for 1 second because I have you just made me think of something. Maybe it's not just a female, but like you're saying, a male feminist, because we're working with another lab too, another male head, he's not the problem. So I had someone email and ask for something from me, and I gave my opinion about these tests and how they would do this and that, whatever, it's not important. And someone responded like, you have no idea what you're talking about. They used all caps. And it was like a senior person who responded and all this stuff. And my boss responded right away and said that was wildly inappropriate, that I have been working here for 10 years. First of all, you shouldn't be addressing someone like they're a research assistant, like look at their title, which is not the point. Like you should treat people regardless of their level. So to be defended and to be shown like you are worthy is wonderful. So I agree. Anyone who supports you.
Yeah, definitely. And then another question, which I think is really relevant, like to me, and I think to a lot of us, especially now in the times of the pandemic, since like a lot of us are spending more time at home, how do you balance your personal life with the exhausting demands of like academia and research?
It's a good question. So during the pandemic, when I was still finishing graduate school, it was really hard. It was almost like we put a timer on because my husband was working from home for the first time too. So it's like you put a timer on and it was like at 5:30 you had to stop working. But it's really hard because you can hear your email and you can, get it on your phone and, you're checking. And, I think the biggest thing with graduate school too, at least I felt that I always had to respond to emails. Like it could be 10 o'clock at night and I would do something. And so that is something I've had to unlearn And you know what? It's not just academia. So the lab that I'm in now is more of like a work-based environment than I think of an academic environment. So academia, you kind of like make your own hours, you make your own days in a sense, right? Your professors, maybe mine was in Tuesday through Thursday and he was home Monday and Friday. The place I am now is a Monday through Friday, 9 to 5 job. And I wanted that. Like that was very important for me. And even in my defense, I remember someone on my committee joking like, oh, you're going to be in the lab 80 hours a week now. It's even worse than graduate school. And I was like, no, I don't think so. Like I made sure I found a lab that was, 9 to 5. That doesn't mean I don't do things on weekends. That doesn't mean I don't do things at night, but I try my absolute best to kind of set like a cutoff, don't answer emails unless you have something due. On the weekends, I do work. I do read articles, but it's typically for my own stuff. So like the grants that I'm working on, the projects that I want to do, that's what I'm reading for. So it's really setting boundaries, giving yourself a cutoff time, trying not to respond to emails at work. But I think that's also a controversial opinion, because there are people that work all the time and they're going to be very successful. But I think that balance, the family life, having a life on the weekends or a little bit of a life is important. Or watching Netflix, I don't know, taking a nap, playing with your dog. Mine is currently snoring under my feet. So
I got my little kitty.
yeah, hello. So yeah, I mean, the best you can, setting boundaries.
Yeah. Yeah, I think that's something I'm learning now. Like I just recently graduated from my undergrad at City College. And I think that as an undergrad, I had a lot of mentors, a lot of people who I would check in with regularly on campus. And I think that now those check-ins are more via email. So like I kind of realized that a lot of my boundaries and a lot of like my push was coming from like my mentors and my like the people who were guiding me. And now that I've graduated, I feel like I have to kind of set those boundaries for myself. Like I don't have nobody telling me like, Denise, you need to do this like right now, because that's like my responsibility now. So I think boundaries is like a really big thing that like I'm now trying to like set for myself in work and personal life and relationships like that. But yeah, thank you so much. What a really super insightful answer. And then I guess I don't know how much time we have. Naomi, do you know how much time we have?
I mean, it's really no like time limit. We're still under an hour, so we have some more time.
I guess another question. I just had another question. It slipped my mind. Oh, okay. How did you know you were ready to go into like your PhD program? Like, how did you like? Because I know that it's like when you commit, and it's like, and I read so many tweets and so many blog posts about people who say like grad school is like a totally different ball game. Like be ready, like you have to be ready. And so, like I said, I recently graduated and I wanted to apply this cycle and I just didn't feel ready. And I think a part of me still feels guilty. Like I feel like why did I not apply or something like that. Like, so I just want to know what made you feel like you were ready, or what was like, I guess that moment that you were like, okay, I'm ready. Cause I'm just kind of like, I guess I'm kind of scared. I don't know when, and hopefully like others can relate. Like, how do I know like when it's my time,
Well, so yeah, it's not for the light of heart, that's for sure. So I did a master's, which was supposed to be, obviously I said that, sorry. So it was a PhD prep program. That's actually how they describe it. And it really was structured. The classes were the same as CUNY classes, just easier. Everything was the same. So I was like, okay, I think I can handle the curriculum. I've always been a top student. I wasn't the smartest in biology, that's for sure. That's why med school definitely never worked out for me. So I knew I could handle the curriculum. I knew I was interested in research. For me, the tipping point was I didn't know what I wanted to study. And, you're competing with the best of the best in a sense, and you're not unique when you're applying to graduate school. Everyone's the same. Everyone's smart. Everyone has experience. So I wanted to be able to convince people I knew what I wanted to do. So that was number one. Number two was I'd been working at NKI as a research assistant for 3 years. And although I had a lot of responsibilities, I was a coordinator for clinical trials, I was doing this neuropsychological rating for all these studies, my opinion didn't really have much weight. And so I had good ideas and I knew we needed funding, and I wanted to be able to, do something. I wanted to go to medical school to help people. So I wanted to do research to help people. And I thought I could do that. But my opinion didn't matter that much. So I knew that if I wanted it to matter, that I had to go to graduate school. So it was really those two things for me. But with that being said, I don't think I had any idea what I was getting into. I mean, like I knew it was going to be difficult. I don't think I knew it was going to be all consuming. So I think depending on if you're going to do neuroscience or if you're going to do something else, I think you need to be ready to commit your entire life and existence. And so I would say the first 2 years were that. Like I had no life. The coursework was so hard in neuroscience, at least in our program, that and it may be something that I didn't know, or that you don't know that I didn't know initially. I read a book about it. It's A minus, B plus, and fail, pretty much. And B plus is not acceptable. And when I mean A minus, I mean A. I don't mean like CCNY, bell curve, like everyone who gets a C gets an A type of thing. As someone who was a former adjunct lecturer, you are getting an A. So you need to know everything, and you have to like smush all this information into your brain. So my first year of graduate school and my second year, I studied and read 24 hours a day, 7 days a week. I stopped exercising. I stopped doing everything because I was a genius that couldn't just absorb information. Like I have to study. So it's all I did. So I'd say for the first year, I didn't see my husband at all. The second year, I gave myself 1 day a week, which was very important because I was starting to mentally go down, and that was not a good idea. I certainly wasn't healthy. So you have to be ready for that. That doesn't mean everyone has to do that. That doesn't mean that's everyone's experience. But I think especially in neuroscience, it's not uncommon to devote your whole being. So if you can be prepared for that, then you're ready. If you can convince people you know what you want to study, then you're ready. And if you have enough experience. So that was the other, or like the third thing I would say when I was interviewing, and I only applied to 5 programs because I limited myself intentionally geographically because I was already established. I didn't want to leave. I didn't want my husband to have to move. I was like, let's just try and see what happens. And so I was told when I was interviewing at Rutgers, which I really wanted to go to, that I didn't have enough research experience. And at the time, I had already been doing research for 7 years. And I'd done animal research, human research, clinical research. So that was a big like eye opener. So yeah. So all those things. So you're ready when you know you can commit your full self, I think.
No, that's amazing. Like that's really good advice because I want to go into neuroscience. My experience and research has been in neuroscience. I've worked at 2 neuroscience labs now. One of them was more like behavioral neuroscience, and the other one was more. My first lab was focusing on the retina, so that was like a little more in depth. Yeah, super cool. So I want to stay in like the neuroscience field, but I do want to go more towards the behavioral neuroscience. And I recently did a research opportunity like with Stanford over the summer. So, and I think that it was fun. It was amazing.
It's cool.
Great experience. But I think that like that really like showed me like how intense it was. Like I was on my computer. It was all virtual, so I was on my computer like all day. Like I just like gross, but I wouldn't shower till like 12 a.m. Like I would just wake up and go straight to it, like because I needed to submit data to my PI and I needed to join the Zoom meetings and the lab meetings. And so yeah, but thank you so much. That kind of reassures me because I know right now I'm not ready. Like I know right now where I'm at, like where I just graduated. I think that I want to gain some more experience, and I think that I want to stabilize myself so that when I do get into something that rigorous, it doesn't affect me so much, like it doesn't drag me down so much, because I know that personally, like things like I get overwhelmed, and so I want to make sure like I'm right here before I can move into something. So yeah, thank you so much.
You're welcome.
good to know, because I've been so stressed. I'm like, oh my God, I didn't apply this cycle. And so
No, please. Whenever it's right, it's right. Like, I don't know if that makes sense. It's you have to be okay with it. So I think the only thing that you're going to notice, so everyone who was in my cohort and is older than me or the same age. So the only pressure that you're going to feel outside is if other people are doing things in their lives, going on vacation, starting families, advancing in their careers, and you're in graduate school. So in my opinion, it didn't matter how old I was. I think I started when I was 27. So I applied when I was 26. At that point, I was like, oh, I'm fine. I can do anything. It's not a big deal. But it was as other people were doing things in their lives that I finally was like, ooh, this is tougher than I thought it would be. That was the only like weird pressure that I think I've said in class before when I've taught, like, you need to think about where you are. Are you okay with where you are and what you're doing? And that's part of being like the mentally ready.
we also have another question in the chat. Marilyn said that you brought up a good point on how many applicants are similar. How did you make your grad school application unique?
I don't think I was unique. Sometimes I feel like graduate school, I always say this to like either students I've had or people I work with who I've mentored. Sometimes there's just a lot of luck involved, right? So you have to have the top grades, count that out. So I think your interviews are really important. And for me, my interviews were not great. I interviewed with one person who sat down and said, what do you want to do for the rest of your life? And prior to coming into that interview, everyone was like, don't say you want to go into industry. At the time I thought about it. It's an interesting way to go, work in pharmaceuticals, do clinical trials. It's very exciting research. And that guy sat down and said, there are no jobs in academia. You're never going to be a professor. What do you want to do? And I was like, what? Huh? And then my second interviewer had gotten in a car accident on her way there and told me she hadn't looked at my CV and she knew nothing about me. So I guess somehow I rocked my interviews. I don't know how. And so that was important. Having a good CV. So, if you have a mentor that you work with now, make sure your CV, shows not just what you've been a part of, but like have a description of what you do, because people will read that. It's not normal for your, CV as you advance in your career to have that information in it, but for applying, that's important, I think. Experience, just get experience. It's better to say, again, I don't know, I'm not on graduate school committees, but I think it's better to say like, I've done this and I learned this technique, but I know I don't want to do that, than it is to say, no, I loved this, and I loved that. So at least if you have like a roadmap, like I did with all these different things, but I can explain why I've tried this, I thought about it, I know I don't want to do it. I love working with patients. I hated working with animals. I like working with people that have Alzheimer's disease and mild cognitive impairment, but I don't want to be a psychiatrist. I don't want to be a clinician. I don't want to, counsel people. So just being able to, show that you've done what you've done and, understand what you've learned from it and what you don't want to do. And that's, I don't know, the best that you can do. Fake confidence. See. I didn't see that there was the light just went out in here. I'm sorry.
Oh, no.
I'm still here. Well, these, these automatic timers.
Okay, we can address this question in the chat, and I mean that could be our final question since we are in that. Oh, okay, I'm glad.
Sorry, the light just went out.
Okay, yeah, so we can just address Michelle's last question, and since we are getting to the hour mark, so that you're good on time. So Michelle asks in the chat, what draws you towards working with the geriatric population?
It's a good question. I've worked with them since I was in high school volunteering. People joke that I'm an old soul, so I really get along well with older people. But I guess, I like working with geriatrics because when I was with this program, I worked a lot one-on-one with people. And so, so you do this test battery. It's an hour and a half long. You do all these standardized measures. People are so nervous because everyone's so afraid they have Alzheimer's disease, right? So your job as the person testing them is to sit with them and make them comfortable. So I would spend the first 5-10 minutes basically distracting people, like when I used to babysit kids, and ask them about their lives. And it was just really fun to learn people's experiences. And it's like, you find that older people, a lot of them are lonely. So, even though you're testing them and even though you're literally trying to figure out what's wrong with them, you're giving them your attention and you're showing compassion and you're learning about them. And so a lot of people just really enjoyed their time. So to me, it's not just like, yes, understanding the heterogeneity of their profile and figuring out what's going on, but it was just getting to interact with them. And they have so much experience and people are so willing to tell you about their experience. And I don't know, you meet some really cool people. And I guess the bigger thing is they're really good participants. They follow instructions. They don't fall asleep in the scanner. They are willing to do things for free and happy because they're helping science. And so it makes life a lot easier to collect data and do research with people who are willing to do it. So that's a good question. Thanks, Michelle.
Okay, well, we are reaching that time, or I think we're a little past that time. So I just wanted to say thank you so much for your talk today, for giving us your time, for giving us your insight. I think that like a lot of us, especially me, like really needed to hear some of these things. And it's awesome hearing from another woman in neuroscience. So thank you so much for all your time, Dr. Reinhardt. And yeah, does anybody else have anything, any final thoughts? If not,